the assembly of subunit proteins of photosystem 1. Okay, how these complicated, complex are synthesized, and how many components are assembled into a functional structure. So it was very difficult to analyze the molecular mechanism of complex assembly, because this process occurs very rapidly. So you cannot follow, okay? It's difficult to follow the assembly process. It's very short time. So we focused on the factor, assembly factor, to find out assembly factors that assist assembly process. Then how this assembly factor works, if we know, we can follow the assembly process. And because this PS1 complex has many subunits, so it was we could expect that there are many factors should assist this assembly. Not only one, more than one, probably many. So today I'm going to talk of several factors we found, but these are not everything. There are many still remain to be found. Okay, so I talked yesterday that this assembly factor had not been found before because probably the strategy to identify the assembly factor is you isolate, for example, the mutant deficient in PS1. But in many cases, most cases, the deficient in PS1 mutant is usually translation mutant, okay, or a transcription mutant, or some mutant lacking one of the important component of PS1. So it was difficult to isolate the mutant deficient in assembly. But as I told you before yesterday, by chance when we were working on hypothetical chloroplast openly framed YCF3 and 4, we knocked out this by chloroplast transformation to know what these two openly framed are doing. Then by chance we found that the mutant of YCF3 and YCF4 or YCF4 deficient mutant completely lacked the photosystem 1. And these two components are not involved in expression of PS1 component. I mean, messenger RNA normally transcribed messenger RNA coding for PS1 component were normally expressed, and translation was not affected. So these two factors are not involved in transcription or translation. Probably most after post-translational events. That means most likely the assembly process. Okay, so I didn't show you this data yesterday. When you look at the immunoblotting data using PS1 reaction center protein antibody PSA-A, wild type is 100%, 10% is the dilution. Okay, and then YCF3 mutant or YCF4 mutant didn't accumulate PS1 protein at all. No, even the 10% level is still we could detect the signal, but it's much less than 10%. So it's almost nothing accumulate. No PS1 accumulated. Okay. So then yesterday I talked But we don't know how this YCF3 or YCF4 act in the assembly or post-translational event of PS1. How? We don't know. So what we did was affinity purification of YCF3. So this was strategy of affinity. We just transformed the YCF3 genes with HA tag. So no wild type YCF3 was present, but this HA tagged YCF3 is present in this mutant. Then we did we purified solved the cytochrome membrane and then purified YCF3, and then I showed you before these two proteins were purified: YCF3, HA tagged, and interacting protein YCIP1. And also very small amount of PS1, but you couldn't detect by staining the polypeptide. But the immunoblotting showed that the PS1 is a little bit present, the PSAF, but not as a peripheral. Then we did pulse chase labeling. Then after pulse labeling, YC3HA preparation contained pulse labeled PS1 reaction center and also PSAF a little bit. But during a chase experiment, this label disappeared. Okay, so that means that the binding a small amount of PS1 associated with YCF3 is transient. Okay, the steady state level is small amount of the steady state level of PS1 is present, but this small amount of PS1 is turning over very rapidly. Okay, that is a so only newly synthesized PS1 associate with YCF3, or YCF3 associate only newly synthesized PS1. That's why this protein YCF3 act in a post-translational event of PS1 synthesis. Okay, this is the way. The same thing, we did the same strategy to analyze other factors. Okay, this is important. So if you understand this, you can easily understand the following experiment. Okay, so just look at the this polypeptide pattern. No, very simple, right? Many minor amount of PS1 and part of reaction center PS1 and PSAF, but not other PS1. So this is Y3IP1. So I skipped this. I talked yesterday already. So our conclusion is YCF3 is interacting with the initial assembly steps of reaction center of PS1. Okay. Then affinity purification of YCF4. No, another hypothetical group of openly different. Of course, this knockout this gene resulting in no accumulation of PS1 like YCF3. So we purified, using after tagging YCF4 with HA, and then purified. Of course, after we isolated thylakoid and the solubilized thylakoid and affinity purified. This is a little bit small. This is better. Okay. So this is a result of purification. Okay. It's a little small. This is A3 is PS1 purified PS1 PS1 complex. So to show the position of PS1 and LHC1 polypeptide on the gel. This is Y type is a negative control. So we use Y type at almost no protein was recovered, indicating this positive HAYC4 is the polypeptide recovered is very specific. Okay. So if you look at this polypeptide, this is a PS1 reaction center here, diffuse band. There are many minor contaminations somehow, but it's some PS1 related proteins you can see. You see? PSA C, E, PSA L, PSA DF, and also some LSC1 proteins also recovered, which means that YCF4 interact not only PSA-A, B, PSA-F, but additional PS1 polypeptide and LSC1 as well. These are confirmed by immunoblotting PSA-A, C, D, F, L, no PS2, almost nothing, and some LSC1. So my question to you is, what this difference of the polypeptide pattern compared with purified YCF3 polypeptide pattern means? No? Why YCF4 associate more PS1 protein, even the LSC1, compared with purified YCF3 preparation? What this means? What you can extract, conclude from this data, or comparison with the data between the YCF4 and YCF3? What you can speculate?
YCF4 is critical for PS1.
Yeah, critical, of course, critical. YCF3 also critical. YCF4 is critical. No, if you knock out, no PS1. But the difference, we observe the difference between YCF3 preparation and YCF4 preparation. This difference, what this difference means or suggests, or tell us what's. Okay, is there anyone? Light harvesting?
YCF4 plays major role in harvesting capsule light.
Okay. Now, LH1 is also present. Why? It's okay. Okay, just think. Okay, so then speculate by yourself, and then I explain. Okay. This also, I did a immunoblotting, pulse chase labeling as we did for YCF3. Okay, the same pulse and chase. Okay, PSAA very strongly labeled in this time, and also PSA C, L, DF. This is YCF4, LSCA1, LSC1, something. So and also doing a chase, PS1, no, disappeared, going down the level. And PSA DF, it's also going down, okay, decreasing. So these PS1 proteins associated with YCF4 are also transient, okay, transient. It's not stably associated with YCF4. The newly synthesized PS1 proteins are preferentially associated with YCF4, like YCF3 also. So that's why we can conclude that the YCF4 is also involved in post-translational event of PS1 accumulation. Okay? And also, this preparation was slightly green. Okay. YC3 was not green. Preparation was not green because probably there was a PS1, but its amount was so small. But this time, no, the protein level of PS1 is more abundant. So then we can measure the absorption spectra of this YC4 preparation. Then, no, CoQ-A, this one. And this part is carotenoid. Can you see it's small? And also the peak of the red region is 678 nanometer. This is the typical absorption peak of photosystem 1. Photosystem 2 is 670 nanometer. So this is not PS2. Or LSC 1 or 2 also a little bit shorter. So this is the typical PS1. The chlorophyll associated present in this preparation is derived from PS1 complex. And the HPLC, high performance liquid chromatography, indicated that chlorophyll A is here. A little bit beta carotene and slightly chlorophyll B. Okay, because it contains a small amount of LSG1, so PS1 core lacks chlorophyll B, but LSG1 contains both chlorophyll A and B. So this represents small chlorophyll B amount, chlorophyll A, beta carotene. Beta carotene is present in PS1 core and LSG1. LSG2 lacks chlorophyll beta carotene, but LSG1 contains beta carotene. What this means? Why this YC4 preparation contain chlorophylls? What this means? YC4 associate not only PS1 proteins, apoprotein, but also cofactor are present here. Okay. This is the same. Right. And then we separated this YC4 preparation on sucrose density gradient to see if the chlorophyll present in YC4 preparation is really associated with proteins or free chlorophyll or not. Of course, the peak of the chlorophyll absorption peak at 678 nanometer indicate these chlorophylls are not free, not bound to PS1. But to confirm, we purified, separated. This is thylakoid membrane, okay? The thylakoid extract, soluble with detergent, and LHC2 band and the PS2 LHC, PS1 LHC1 band is separated. And HA tag to YC4 preparation, we see here and here, the green band. So indeed, this shows that the chlorophylls recovered with YC4 preparation is large, in large complex. And the position from the gradient is very close to the PS1 LHC1 supercomplex, and this is PS1 core complex. Because there's no PS2, so PS2 core is separated here, and the PS1 core is separated here, but this contains only PS1. And then we analyze the polypeptide of this band. The Y3 band is colorless, and also immunoblocking. To analyze more in detail the composition of polypeptide. So it's a little bit difficult. Small, so it's complicated. This is a reference of PS1 LHC1. So PS1 LHC1 polypeptide pattern here, and Y1 here. Polypeptide PS1 reaction center, This is YC4, HA tagged YC4, PSA D, L, E, C. Okay. And Y2 green band that is heavier and larger size, PS1 reaction center, D, L, E, C, small PS1 polypeptide, and LC1 was there, associated with PS1 core. That's why Y2 is larger than Y1, because the presence of the LSC1 association. And Y3 is colorless and contain only YC4. The only YC4, but still so large, so that means the YC4 forms the oligomeric forms, large oligomeric oligomer. Okay, is that clear? And the immunoblotting also show that Y1, this one, contain PSA A, C, D, E, F, but not G, I, not K, L, no LSC1. But Y2 contain PS1, PSA A, C, D, E, F, I, L, and LSC1, LSCA 1, 2, 3, 4, 5, 6, 7, 8, 9. So LH1, almost all LH1, but the LSCA2 is very low. 9 is also very low. So LH1, at least 7 different LH1 are present. Okay, now you understand what's the difference between YC4 and YC3 preparation. So YC3 is involved in initial assembly of PS1, particularly assembly of reaction center protein PSA A and B. Okay, okay, and then YC4. This is YC4. YC4 is required for integrating other peripheral small PS1 subunit to form a PS1 core complex, subcomplex. We call it subcomplex. And also, YC4 is involved in the associating of LHC1, LHCA1, 3 to 8, that are located in a 2 layer of the tetramer on PSA F and F side. I showed the day before, yesterday probably, the structure of PS1 and the LSC1 tetramer. For LSC1 tetramer, there are 2 layers on one side. Okay, but there was one dimer, LSCA2 and 9 were present here, this side. Do you remember that? So in the YC4 preparation, lacks LSCA2 and 9, no, only little, very little. And also here, PSA K and PSA G are present in the edge of the 2 layers of tetramer. That stabilizes the, but these 2 are still absent. So PS1, LSC1, subcomplex. It's not completed. Okay. So our conclusion is YCF3 is involved in initial assembly. The YCF4 is required from initial to the almost the final assembly process, all the way from beginning to the end. And then finally, dimer of LSC1, LSCA2 and 9 associate this position, and PSA K and G are present here. That's required for stabilizing LHC, no, 2 tetramers of LHC1, no, this side is integrated. Okay, so these 2 factors are very essential for this process. And from our data, we could make this working hypothesis. Still my hypothesis, probably. But our data is consistent with this model. But it's not easy to detect this assembly intermediates biochemically because this process is very rapid. And if you try to look at this assembly intermediate using a wild type chloroplast, bunch of mature PS1 among the huge amount of PS1, mature PS1, only small tiny amount of assembly intermediates are present. So this situation is difficult, right? For example, if this is 1%, so 99% is mature PS1 and only 1% you have to find. It's difficult in a biochemical method. Okay, so this analyzing through this assembly factor was so far rather successful, and I think we need to find out other new assembly factor to completely understand this assembly process. So this the data was this experiment was done by Sridhar Nirpari from here. He was a postdoc in my group some years ago. He finished his PhD here in Hyderabad and then stayed several years in Okayama. And he did this very excellent work. And another interesting factor was CGL71. This is a chloroplast green lineage 71, number 71. This was also a nuclear encoded factor, also PGY, pale yellow green mutant in vascular plants, Arabidopsis. And also this is also present in, this gene is present in the shell bacteria. So it's conserved in a photosynthetic organism, so it must be very important. So this factor was first reported in shell bacteria, mutant, and also it shows that slightly PS1, the amount of PS1 decreased. But in Arabidopsis, significantly decreased. So this factor may be involved in PS1 accumulation, but we didn't know how this works. So before we starting Arthur Grossman in Carnegie Institute in San Francisco, California, analyzed this CGL71 mutant. And they concluded that this may be involved in PS1 assembly or post-translational level. And also, interesting phenotype is white type. If you grow the cells on agar plate, white type like this, and CGL71 mutant in oxic condition, in the presence of oxygen. This is a photoautotrophic condition. The cell didn't grow. But anoxic condition, it grew photosynthetically. So somehow this mutant is sensitive to the presence of the oxygen. And then PS1 works well in the absence of oxygen. So PS1 must be very sensitive. So what we speculate is what the oxygen affecting PS1 is iron sulfur cluster. PS1 contain 3 iron sulfur cluster or iron sulfur centers that works as an acceptor of electron acceptor of PS1. Stabilize the charge separation and donate this electron to ferredoxin and finally to NADP plus to produce NADPH that is used for reductants for carbon dioxide fixation. So from this phenotype, we can speculate, somehow acceptor side is damaged in this mutant in the presence of oxygen, but it's stable in the absence of oxygen. So first we analyze the polypeptide accumulation if this presence of oxygen is affect specifically to PS1. So this we tested only PS1 here. A wild type dilution 10, 100, 50, 30, and CDL71 grown in oxic condition. So PS1, PSA-A, C, D, L, subunit are almost missing in the cells of this mutant cell in the presence, grown in the presence of oxygen, but hypoxic. So it's low, very low CO2, O2 condition, nearly anaerobic condition. PSA-A, C, D, L are accumulated as expected from the growth in anoxic condition. Okay. So PS1 is proteins are unstable and in the presence of oxygen and stable in the absence of oxygen. Okay, this was confirmed. So we did the same strategy to tag CGL71 with HA and to purify to see which polypeptide interacting with CGL71. Okay, the same things. This is purified. There is no negative control data, but negative control data is the same, right, as YCF4, YCF3, no white type. So we know that there's no almost very low non-specific no recovery of polypeptide. So we found only CGL71 like this. We didn't see any very major proteins interacting, but we tested the immunoblotting data to detect very high sensitivity of the small amount of PS1 protein, whether the PS1 protein is interacting with CGL71. The white type, almost nothing. And the CGL71HA, we could detect slight amount of YCF4, YCF3, Y3IP1, and PS1 reaction center, but not other small PS1 subunits, PSA-C and PSA-D and E. Why we focus on this is PSA-C associate two iron sulfur centers, FA and FB, as a terminal electron acceptor. So we just suspected that this might be interacting with CGL71, but was not the case. And also PSA-D and E are also on the peripheral stromal side. PSA-C was covered with these two subunits. So, but these were not interacting. But only reaction center we found interacting. And reaction center contain one iron sulfur cluster, Fx, that accepts electron before Fa and Fb. And just present is between the two reaction center PSA A and B. So maybe this protein is probably involved in stabilization or protection of Fx rather than Fa, Fb, Fx to protect Fx from oxygen. And also, this is power state leveling, and it's not so clear. CDL71 here. And reaction center proteins, this diffuse band is not so strong signal, but it's really newly synthesized reaction center protein worth interacting. Okay. And this is also interesting. CDL71 is turning over during a chase. This protein is turning over, like YCF3 also turning over. YCF4 was not turning over rapidly. So these two are very interestingly, very rapidly turning over. Okay, so this is also three published. He's mainly involved in this work with the collaboration of Arthur Grossman's group. So from this data, we speculate that when the two reaction center PSA A and B proteins assemble in into the cytochrome membrane, as I mentioned, YCF3 and Y3IP1, two factors were involved, and also CDL71 is required. So probably before the on the stromal side, PSA C, D, E assembled. Before here, FX is assembled on stromal side of this region. But before the PSA C, D, E assembled onto the reaction center, this FX is exposed, could be exposed to the oxygen. So CDL71 bound to this region. This is a model to prevent the access of oxygen to this iron sulfur cluster to protect. No. And then PSA D, E, C here is assembled. Of course, in that case, CDL71 is removed, exchanged. Then once this assembled, Fx becomes stable because oxygen will not access to this site. Okay, so CDL71 is something like this, where it's a kind of dimer, it's a model, and oxygen is there. So iron cluster center is assembled. Then this bind here. This contains 1, 2 transfer helices, so it's covered like this. No, that's the model, working model. So no, it's once it's this PSA C, D, E is associated, it's replaced by this. Also covers this side, right? So the working hypothesis is the same things, but it's CDL71 also works this region, these steps. No, Arabidopsis is PYG7. Chlamydomonas is CDL71, and cyanobacteria is YCF37. It's a different name depending on the different organisms, but because of this, it reflects the mutant phenotype, pale yellow green without PS1, and Arabidopsis doesn't grow without photosynthesis, so it becomes, the seedling becomes pale yellow-green. Cyanobacteria still can grow without this somehow. PS1 level is reduced. It's always the case. Some phenotype is weaker in cyanobacteria and stronger in vascular plant. Okay, so this is a almost. So this is what we have for the moment, and also there are not so many other factors are found. So this I talked about 4 different factors. 2 factors are associated with each other. So somehow major part is, I think, these 4 factors I talked is very major assembly factors. You can see CDR71 contains small amount of YCF4, 3, Y3IP1. So it's separated mostly, but YCF3, YCF4, Y3IP1, CDR71 are mostly closely associated. But if you purify, the interaction is not so strong, so it's separated. But small amount is present. No, negative control has nothing. Okay, so this is not. We don't think it's contamination, but it has some meaning. Okay, so we have no data, but another interesting or important question to be answered for PS1 assembly is how cofactors, see, chlorophylls, carotenoids, and also as a acceptor, there's a naphthoquinone are also present. These cofactors are how cofactors are integrated into PS1 complexes. This is totally remains unknown. This is also very difficult questions. And we try to analyze this process, but so far we didn't have any fruitful results. But in the next lecture today, I will talk about a little bit about coupling between the chlorophyll synthesis and also subunit assembly. Okay, so I'll stop here, and also very much welcome your questions or small c. Here, C071, HA. Which one? And where? Oh, of course, this is a nuclear encoded, so CD71 HA is fused to a C terminal side of CD71. This is nuclear encoded. But in the case of YCF3 and 4, sometimes we made both fused either C terminal side or N terminal side. We made at the same time. And sometimes if you fuse to C terminal side, the protein may become unstable, or N terminal side may make a protein unstable. So we did both at the same time to save time, to see which is better, or which is better for purification, or which is better for the stabilization. But in the case of the nuclear encoded protein, we always fuse at C terminal side. Okay. This is just a strategy. it's not very important meaning, but sometimes if C-terminal fusion at C-terminal side didn't work, so we have to find out a good position in the middle of protein. But that is not easy, right? So that's the case, okay? Because these data are so tiny detailed, so it's maybe sometimes difficult to follow. But it's if you have which data, well, it's difficult to understand. If you say me, that helps me to explain better. But okay. NHC2? Which? Hmm? Here.
Yes.
Okay, this is thylakoid membrane. So Chlamydomonas has very several LHC2 genes, but some are very, very similar each other. So we categorize major LHC2 as LHC2 type 1, type 2, type 3, type 4. Okay, so this is type 3 and 4. Yeah, it didn't separate, but this position LHC2 type 2, 3, and 4. This is LHC2 type type 1 here. It's hard to say type 2. No, type 1. Type I think type 1. It's separated somehow. I don't know. Probably one of this one is type 2 and this one is type 1. Sorry, it's my mislabeling, but anyway, these are all different types of slightly very similar each other, but it's a different type of LHC2 apoprotein. Okay, so this is thylakoid. You can see from this part. Okay, so I have a question to you. After pulse and chase labeling, the pulse labeling, if you look at this data, could you say something interesting to you? No? It's important to see, if you see the data, wow, this part of the data is interesting, or this part of data is strange. This impression, right? If you look at the data, raw data, this is important for if you do a research. You get some data. Okay, I got the data. That's all. Go to have a coffee. Is that right? No, if you see the data, try to find which part is interesting, which part is unexpected, which is new. It's better to spend some time to speculate. So this is not your data, but if you look at this, you can try to find something you feel unusual or strange or interesting. Is there anything like that? Try to find something you impress you. Anything. Do you have something? After purification, she said strong signal was found in the label for that signal, but we couldn't detect any signal in thylakoid membrane. Why? Okay. Oh, that's good point. Is there anything You think strange or unexpected or want to know more further? Do you have something? Yeah? Tell anything. No? So, many protein? Some proteins here, which you are impressed, strong. Which one? Second? D1? Why D1 is very strongly labeled? That is a good point. Something like that. You should know. Just see. There are many bands. That's okay. Complicated. I don't like. But one step further, you should go. Then some bands are diffuse, but some bands are stronger. Some bands are weaker. Something like that. That you should notice to find the new things. Otherwise, you miss the data. And then you bring to your supervisor. I get this data. I don't know this is interesting or not interesting, successful and unsuccessful. I don't know, but supervisor asks supervisor, what do you think? This is not good. It's not interesting or it's not fun. After doing an experiment, if you find something interesting by yourself, that becomes science. Okay, it's not. If you just do experiment, that's a technician's work. Okay, but if you find something from the data, or if you try to explain something from the data, that becomes scientist. Okay, so question first: why after purification strong signal detected CD71, but not in the thyroid membrane? This is very important. This affinity purification so excellent. You can concentrate significantly, so signal becomes so strong. But here, the amount of CDL71, catalytic amount, very low. So this should be signal, but you couldn't detect because of the sensitivity. But if you detect strongly, so other protein also appearing. So this signal may be covered by other signals. So that's why it is strong signal, but okay. So another interesting suggestion is why D1 is strongly labeled. There are two possibilities. One is this is not labeled with carbon 14, but it's sulfur 30, S35. So if the protein lacks methionine and cysteine, you can't label. Okay, and also if the protein has a bunch of cysteine and methionine, it's strongly labeled. But this is not the case. D1 is another possibility is protein is abundant and synthesized very rapidly. Okay, so then you will get strong signal. Or the protein is not abundant, but it's turning over very rapidly. So it gives you a strong signal. In the case of D1, the amount of the protein is much, much lower than LHC proteins, but still strongly labeled. And D1 has not so many cysteine or not so many methionine. So this is, as you know, D1 is turning over very rapidly under illumination. So if you don't illuminate the cell during a pulse labeling, D1 is normally labeled very like other proteins. So this is strong. And LHC is very abundant, most abundant proteins, but labeled strongly relatively, but it's still compared to the abundance of LHC2 protein in the thyroid membrane relatively. So the synthesis of LHC2 is not very high. And also these are two bands here. So this is ATP synthase alpha beta terminate. This is also somehow always strongly labeled. That is the case. Okay, PSA B, not really. You don't see in the thyroid membrane. It's not, synthesis is not high compared to PS2. PS1 and PS2 accumulation level is nearly the same, but PS2 is more strongly labeled. So PS2 turnover, PS1 doesn't turn over. Okay, that is how to see the data. So now if you do your own experiments, so after getting the data, you should take time to see carefully data. And then think about from which what you can extract. This process is very important. Okay. Any other questions? No? If not, I stop my talk.